So can you see my screen?
We can see your PowerPoint.
The whole screen,
with all the PowerPoint controls.
what?
But we see your PowerPoint window with like some slides down the side, and controls and that sort of thing. It's not full screen.
Oh, stop sharing, reshare again. So how is it now?
That looks great. It's full screen now.
Okay. So I'm using two screens. One of them is old, so I stopped. Now I can just see my own screen. My name is Shaza. Yeah, and I'm so happy that I got to present at this. And basically, I'm working on a spheroid-based model. It's under my research project is a spheroid-based model and a preclinical model of triple-negative human breast cancer cells. And it's about unraveling the effects of vitamin E from palm oil. It's under Food as Medicine research strength under the School of Medicine of Jeffrey Shaya at Monash, Malaysia. So generally, I'll give an overview about the project, some of the research results, because I have a lot of results, and discussion, which I'm really looking forward to your feedback about it, and what I'm planning to do next. So my research is about breast cancer. So breast cancer is one of the common female cancers along the globe in more than 200 countries along the world. In Malaysia, it's the most common one and account for about 17% of the total casualty of cancer in Malaysia. So generally, breast cancer is breast cancer is a heterogeneous disease. It presents with different types, and it can be broadly differentiated into 5 or 6 subtypes, depending on the tumor, lymph node, and histological presence, plus the biomarkers. So generally, luminal A, we have luminal A and luminal B, and it has the common 3 markers, which are estrogen, progesterone, and HER2. We have HER-enriched subtype, and all these three types have been easy to treat in a way, as long as it's caught early, except triple negative. Triple negative is very hard to treat because it doesn't have targeted markers. The breakthrough in treatment of breast cancer is that the presence of targeted markers that can be targeted by monoclonal or chemical treatment. So triple negative is one of the hardest disease, our breast cancer disease, to treat because of lack of approved targeted therapies. So generally, when we start any new treatment, if we're looking for targeted therapies or anything, the research starts by cell lines and animal models, and then it proceeds into human. So cell lines is one of the most common basic preclinical research that is used to treat, to check and investigate the efficacy of different drugs. So although the 2D cell cancer is used widely, one of its problems, although it retains most of the markers that has been found in vivo or in the patient, it doesn't represent the whole environment that it might be inside the human body or in vivo. So for an in vitro model to be used in a better way, of course, I mean, nowadays organoids are much better because they come from the cells itself. But second best can be that we form the spheroids, which are a spherical shape of the 2D cells. They I'm sorry. So it's the monolayer cells or the 2D cells that aggregate towards each other. When they aggregate towards each other in certain setting, they will form different types of cells and will cause different gradient of these cells. So as seen here, we can see that if we can form the spheroid or the globe that of these cells, we will have some differentiated cells that we can use to check the efficacy of the drug at multiple levels and to some way recapitulate what's happening inside the human body. So it's usually when it happens, it will be compact. It will have an inner core of apoptotic cells, and we'll have quiescent cells, which are dividing cells, dividing cells that are not yet dividing, like in stasis. And then we have the outer layer, which are made of dividing cells completely, which is called quiescent proliferative cells. So generally, the spheroid model can be a good model to test breast cancer and can recapitulate some of the in vivo characteristic and can create certain challenges for the drugs to go through it. So because there's a metabolic gradient, there's also an oxygen gradient in it. So that's why we now, when we try to check the efficacy of vitamin E, we try to form an acceptable model of a spheroid that can tell us a little bit about what happens when this drug is killing the cells or causing antiproliferative effect. So generally, we tried different types of models, but the model we went with is the last one is made with liquid overlay from MDA MB468. I've tried another cell line, but I'm not going to present it here, another triple negative cell line. And this is how it looked like. So under the bright microscope, we can see like a darker space here, an outer layer, which we will think is the proliferative area, and then the quiescent area in the middle. We use the HS immunofluorescent to check the DNA, to check the DNA, to detect the DNA and to detect the proliferative state. As we can see here also, it's showing us multiple light areas, which will show more division of the cells with a bit of darker side inside. We examined the cells under the field microscope to see the outer layer, how it's made and how it looked like. So generally, we can see that there is a rounded structure shape, which is the spheroid. And then we are at higher magnification, we can see the folding of the membrane and the intersection or the interconnection between the cells and so. And later, we examined how the genes are the stem cells and how it's reflecting and how much hypoxia is inside. So we went for CD44 and HEV alpha to check the gene profiling of this model. So palm oil or vitamin E, which is from palm oil, is a natural compound. It contains tocopherol and tocotrienol. It's made of different isoforms, alpha, beta, gamma, and delta. So, in general, it's very hard to get a simple one separately, but generally the tocotrienol extraction, there is a form of tablet that we use and which we tested, and we tested also the one which is purified from the company itself or the producers of it. And so it contains 75% of tocotrienols and 20-25% of tocopherols. It has been reported in 2D cells and in animal models that it can be can be helpful in non-communicable disease like hyperlipidemia, hypertension, diabetes, and is cytotoxic to certain cancer, such as breast cancer, lung cancer, brain cancer, prostate cancer. And there were reported different ways by which this vitamin or TRF or tocotrienol-rich fraction can mediate its effect can cause reduce in cell cycle, induction of autophagy, death, can inhibit cancer stem cells, and also can induce apoptosis and decrease angiogenesis in cancer cells. So after that, we tested the TRF on 2D cells and on 3D cells. As seen here in 2D cells, we can see there is time dose reduction at 48 and at 72 hours in the proliferation rate. We use cell proliferation cell titer glue to detect the ATP production and emission. In 3D model or the spheroid model, we found also the same pattern, but there was some resistance as expected because it needs time to work. At 72 hours, still we got the IC50, and after that, under the microscope, as seen here, this is my control, which is the vehicle. The spheroid cells, we can see it shows some of its characteristics. And then at 10, it's decreasing until at 100 total disseminated, we cannot see the whole features of the spheroid. So for further investigation, we did liquid chromatography mass spectrometry, LCMS. We quantified the control and 2D and spheroid culture, and we checked the upregulated and downregulated protein. Overall, a lot of proteins came. The one that passed were more than 200. In this talk, I'm going to present the differences that we found, the differentially regulated proteins that were detected between 2D and spheroid culture, which were in total were 54, and then between TRF treated and untreated spheroids, and paclitaxel was used as a control. So generally, when we use when I analyze the data, we found that most of the, I'm gonna go back to my notes. So we generally, the difference between 2D and 3D culture or the spheroid culture, we found that most of the proliferation was more, pathways were more activated, such as the cytoplasmic ribosomal protein, the endothelial, the VGF also was upregulated, glycolysis, and other proteins that are involved in cell growth and proliferation. Along with other proteins that are approved and are still in research or in clinical trials that considered as triple-negative breast cancer targeted for targeted therapy. So paclitaxel was used as a control. So overall, it showed some of the pathways of how the paclitaxel worked. So the tubulins pathways were decreased, and it didn't show the whole formulation of how paclitaxel worked as expected, because it recapitulated, cannot give us the whole picture about what will happen inside inside the human, but it gave a general idea about how paclitaxel pathways are working. Paclitaxel is a known triple-negative chemotherapeutic disease, chemotherapeutic treatment. So in TRF, using tocotrienol, only 4 were upregulated, and they were mainly for cytoplasmic translation when we did the ontology, the gene ontology, and cytoplasmic, they were mainly in cytoplasmic translation. The downregulated by them, they were mainly pathways that kill cancer, as shown here, and are involved in disease regulation of cell cycle, apoptosis, and other pathways. So in general, several proteins were differentially regulated between spheroid and 2D culture, supporting the transformation of 2D culture into more complex structure. And most of these proteins, as I said before, are component organization of development and communication between cells easily. A number of these overexpressed proteins found in vivo and considered potential. One of them is the PGK and HP90B1, which I think in the first phase of clinical trial. TRF itself inhibited the proliferation of triple-negative multicellular tumor spheroid. The half maximum inhibitory concentration was established. Several DEBs were involved in TNBC survival were downregulated by TRF, especially those two. And some of the pathways is the RHO GTPase pathways, which is involved in motility, invasiveness, and metastatic in TRF. So these are my next steps or advantages and disadvantages of what I did. There is no, I would love to identify the ultrastructure of the TNBC and see exactly how big is the necrotic part, how big is the quiescent part, improve the imaging techniques, single cell RNA sequencing if possible, use spatial omics method and analysis with lab confirmation of the identified protein enrichment pathways. I acknowledge my supervisors and thank you. I'm sorry, I was very fast.
So does anyone have questions?
Yes, I have a just an odd sort of question with those, I guess the volcano blocks. There's a strange pattern to them that I haven't seen before, the sort of parallel wobbly lines.
Oh, sorry, I can't hear you very well.
Just something I hadn't seen before with the volcano plots is that sort of pattern of curved lines in them. Just wondering what that comes from.
Can we flick back to the volcano plots?
Which one?
Yeah, so that one on the left, for example.
Okay.
Yeah, I don't know, maybe this happens in mass spec, but it's a strange sort of pattern of curvy lines.
Okay.
How that happens.
Yeah, usually we don't. In, say, an RNA-seq experiment, you wouldn't see any lines like that, except maybe at very low numbers.
Yeah.
Very low counts.
Yes. So in RNA-seq,
the volcano is not for the protein. It's actually the pathways that generated by the proteins.
Oh.
Yeah.
Oh.
So different pathways according to the FDR on the side, and this is the different pathways. because I did
Oh,
mass
okay.
spectrometry, not single cell. So I have no idea which protein is where exactly.
Okay, right, okay. Now that makes.
Does it make sense now or no?
I suspect it will make more sense if I think about it more. But yes, okay. In general, yes, okay, yes. That's not what I thought the plot was. So okay, yeah, cool. Thanks.
I'm really looking to feedback and criticism, 100%. My background is zilch in bioinformatics. Everything I knew is during my PhD, and I'm honored to share it with you. If you can have any input, I'll be happy to.
Well,
Shaza, oh yeah, sorry, go for.
well, maybe basing a more general topic. We have these organoids with well, I guess a fairly straightforward three-dimensional structure. So, do you have plans to investigate the 3D structure of the organoids and maybe the effects of your treatment are localized to the outer part of it or the inner part of it or something like that?
I was lucky enough last year, I came to the organoid program. We investigated the TRF. So far, only we got the IC50. It's killing, but still I'm waiting for the rest of the investigations. And of course, there is always the money problem. Some of the problems with my project is that I started it during COVID, and I did multiple optimization, and by the end, most of the plates come from the US, so I had to wait for them. Then my PhD finished. But I was lucky enough, I ran some testing under this is where I met Shani. I don't know if she remembers me or not, but I was lucky enough. I sat at one of her presentation, and this is when I knew about the spatial omics.
All right.
Yeah.
I mean, I guess thinking about approaching spatial transcriptomics on this tissue, you could take the approach of getting a thin slice and doing one of the 10x style spatial techniques on it, but I do wonder if you would get similar good enough results from simply taking a core or a slice. You don't, because you might expect that your circular slice through an organoid is mostly symmetrical. You could then just take a radial segment, right?
Yeah.
And then sample along the radial segment with bulk experiments. It's a little bit like what Murata was doing with the mouse heart stuff, and it might be giving you similar sort of information to what you will get. May not get, I guess you may not get sort of cell resolution niche information that could be interesting.
Actually, it's pretty tough. It's very easy for me to cut it. Once if it's fixed, then it's very easy to get a transaction out of it, the whole thing. It's just after I treat it, if you notice here, if I go here, it disseminates, especially if I go until the IC50, it really disseminates under the microscope. Yeah.
So after treatment, it becomes a lot more fuzzy.
Yeah. So
histological term.
At higher magnification, you can see single cell, but the microscopes I'm using here, they go up to 40. So this is what I get.
Yeah, I see.
40x.
And in that case, taking a representative slice might be harder. There might be interesting details in the protruding bits, I suppose, with more detailed spatial analysis.
Thank you so much.
I guess the other thing that I'd imagine people would be interested in an organoid like this is comparing to existing data or samples of the real organ, or in this case, the real
Yes.
breast cancer, which I'm sure there's tons of public data already to compare to as well. Try and establish what cell types you're finding and whether they match what's in other data already. Did you have a particular plan? You know, you gave a list of the things that you want to try. At the end.
Yeah, I do a lot. I mean, I would love to see how big is the core, and I'm intrigued clinically. Is it affecting me? I want to see where is the downregulated are happening. Are they happening at the core? Are they at the outside? Are they in the inside? Is it, say, if I compare it to the organoid, is it a solid clinical model that we can use? And how differentiated, as you said, in single cell sequencing, would we see differentiated cells? Although when I run the genes, they're quite different. I mean, according to which procedure I use, or which procedure I use the scaffold, which use a rigid, and then they are showing different upregulated and downregulated genes than the other ones. So it gives you the idea that maybe the molding of the cells will decide what will happen. And most of these cells, they formed after I had an ECM of collagen 1. So it needs some sort of a matrix to solidify it and make it as you have seen it here.
It'd be certainly interesting to see whether the markets that also respond to TRF are only in those surface and surface proliferating ones, or whether there's something happening all the way, you know, gene regulation changes happening all the way to the core.
Yeah, true, correct. So thank you so much.
Further questions on Zoom?
So anything in the chat?
Did you ask me a question? I can't hear it.
No, we're just checking that if there were any questions on Zoom, any extra questions on Zoom. I think looks like everyone's satisfied. It's good. Thank you.
Well, like, I guess it's probably other people around the place doing things with organoids and spatial.
Yep, Alex Coombs is definitely doing a lot of kidney organoid stuff. And is it Helen? Was she doing that? I know Stuart was doing some more stuff. No, remember? Yeah. Alex is very keen on is doing kidney organoid work, spatial kidney.
And I guess it's like most of the spatial stuff, there's tissue. How do you replicate results with the organoids? In other words, it seems possible to get some actual something like a replication of multiple organoids doing the same thing. It's nice.
There was a study that I saw talked about, I don't remember who it was. Was it at Abacus 2 years ago, where they had real trouble getting reproducibility with it? They were finding that the results were very dependent on whether they travelled in an esky from year to year, you know, whether they were put on ice and transported or not. And it was very hard to replicate. Oh, there's a COVID infection. infection model, I think. Coming back to me, I don't remember what it was. Which made me, think it could be really tricky or need replicates to come up with things that are maybe less essential with spatial experiments. You can see the niches in rather than relying on a bulk experiment where it's all mashed up and the variation you might feels like you might be able to get away with less replicates. Does that make sense to you?
Michael C. Like our Michael.
Oh, maybe it was.
I'm pretty sure it was.
Yeah, there you go. Type in something else. I don't know of any work yet because I'm not following organoids, but any work of how far people have gone to look at cellular structures or niches, let's say, by spatial transcriptomics or spatial proteomics, and seeing whether they also exist in the real organ. Because you're not necessarily replicating all of the detail of the original organ in the organoid, but you might still be replicating niches that exist in the organoid. You know what I mean when I'm talking about niches, like particular arrangements or concentrations of particular cell types that tend to be near each other in the same sort of proportion or arrangement. So that I guess it's less difficult with a breast cancer model because they are a bit more like proliferative blobs. So maybe actually this is possibly closer to the real thing than some of the other models, I suppose.
So that'd be like the pattern you had with the organoids of a proliferation on the surface and the aquatic in the core. Is that a good model of actual breast cancer?
Pardon me?
Yeah, so I think you had a diagram with like the organoids, like they're dividing, the cells are dividing at the surface and then they're sort of necrotic at the core. Is that a good model of breast cancer? How it goes?
It resembles the avascular metastatic part. If you check it outside, then you will find it resembles that in theory and what I read. So that one is not connected to the blood with vessels and everything, but it's moving into the blood. So we wouldn't know how much nutrition it's getting, but we already know the outer layer is working, the inner layer is, because it's a solid tumor. So it's expected that the middle is necrotic by the time it's metastasizing. And correct me if I'm wrong, if anyone is working in breast cancer.
So in this model, do you see something like metastasis occurring? Is that the diffusion that you're seeing as well in your images here? Are they potentially metastasizing cells, or is that something different?
To be fair, once I submit, I'm planning to stain. So by that time, I will know what's coming out. But meanwhile, under 40x, it looks like the outer side. I mean, if it's a bigger magnification, I can go here. So this outside, instead of being the proliferative part, is not being solid. So if you're not, this is my day 6. So this one is actually benign after production development of the spheroid, the control one. So still it's intact, outer layer. But once I treat, then it's not intact. I mean, if I maximize or if I stain with the HS, you can see the outer layer is very clear, still positive and the control. So what I think is just is breaking out. That's why, I mean, when I have here Dr. Anita at the lab, is that you can present it to the people who are working with the spatial omics. We will know what's going on, what's coming out. Is it the necrotic part? Is it the outer part? Or is it just mechanical? Which I don't think so, because we have DEPs which are upregulated and downregulated after treatment, I mean, in comparison to the control, plus the data from previous research is done on TRF.
Yes. Probably. So are there many free cells you think finding their way into the supernatant? Cells that are completely separated?
But I'm sorry, I missed the first part with some deep, yeah.
Are there many free cells that have completely separated from the main organoid and are free in the supernatant or free in the media?
When I leave them more, when it's not treated, I do find the smaller size ones. Yeah. So when I did the electronic microscope, they were smaller size ones on the side. Once it grows, when it's fully developed, some of the cells will escape, because I start from high up. The idea is that to form it, the MDF468 cells, they by nature they need some sort of a scaffold to hold them. So I started by hanging drop to initiate the aggregation of the cells, then I move them. So if my technique is not, I don't know if it is my technique, which I doubt, because when I look at the cells, they form. But after day 6 and 7, the control itself has started giving smaller cancer cells. So, but that is the untreated with TRF.
I see.
With TRF, I didn't see any on the side.
I guess where I'm leading with that thought is whether the free cells would be metastatic type cells, or whether that's just, you know, it could be a naive understanding about how tumors work, and that's not what's happening, or you know, organs just fall off and become detached and they're not really metastasizing. But my thought process is leading down this, you know, okay, if you could take the media and look at that again, not necessarily in spatial, but bulk, is that going to have an interesting profile that tells you something about metastasis? Or it might be nonsense, because it might just be dead cells or RNA or something else. I don't really know.
Yeah. I'll give it a thought. Maybe I'll even stain them and see what they look like. Maybe it's just metastatic cells. Could be.
Interesting.
I am mad that I missed this. I'm sorry. Because I wanted to see how did you manage to get, because breast cancer organoids can revert cells and then have cells that are not cancerous anymore within the same organoid. I wanted to know how you deal with that. This has been discussed?
You are asking about how it's developed?
So within the organoid that is the cancer organoid, the one that you started with cancer cells, there has been experiments that I have worked with within the same cancer organoid, the organoid that derived from cancer cells. Some of the cells ended up being normal cells without producing tumor. So I wanted to know how to work with those.
Unfortunately, my model is not from primary cells like the organoids. My model is from the immortal cells, the one which are kept actually. But it's believed they have inherited their, right, because they are growing from stem cells also, and they can form stem cells in 2D culture, as proved by other researchers. You can find CD4, you can see CD24, And from these stem cells, we are developing the spheroid culture, which is in a way a bit easier to develop than organoid culture. I think also cheaper and easier if your lab is not fully developed for this, and a better model to study the disease, because most of the 2D culture is flat. So once the treatment enters the body, it has to go through a lot of barriers. One of the barriers can be created by this model, which is, I don't know if you can see it. So it has, once it's developed, then the spheroid will have oxygen gradients, will have nutrition gradients, and then I can add to the outside. Some researchers even add fibroblasts and other in the ECM, in the extracellular. Plus, you will have interactions between the cells. So it's not easy for the treatment, it's not fully distributed at the same time. I mean, equally distributed, it's just distributed from in to outside. So as like, and then after that, we'll see if the proliferation, say, if there is no morphological changes like what I got, then I'll check by using proliferative agent to check the ATP and the Vincent or other agents to check how is it dying, is it living, is it increasing it or not, in comparison to my control. I hope I answered the question or not.
This is a bit different of what the organoids that I want to working
it's
with.
quite different because it's from immortal cells, the 2D cells, yes. And the organoid people, you can correct me if I said something wrong about the organoids.
There's no more questions. Yeah, so yeah, thanks again, Shaza, for presenting your work. Interesting to talk about organoids. Yeah, it's fine. Yes. See.
Thank you.
Thank you. Thank you, Shaza.
Thank you.
Stop.